Hello, this is Ed Chapman, and this video cast is going to be about how microscopes work and how we use them, focusing also on the different kinds of microscopes that are currently available in different types of labs. I'm hoping you guys recognize this guy's picture. This is Antony von Leeuwenhoek, and he is the inventor of one of the early microscopes. I'm sure he would be very impressed today if he saw our microscope equipment because they are much better. A lot has changed in 400 years. Okay, here's two pictures of the first two microscopes invented in the 1600s. On the left here we have Robert Hooke's microscope, which looks a lot like a conventional microscope. You put your eye right here, you look into the eyepiece, and down here somewhere, I'm not exactly sure where, goes your sample. So let's say your sample is right here. This situation over here is a flame. So you get a flame here. This is the oil for the oil lamp. And I believe this is a ball of water. This is a globe of water that focuses the light and sends it to this lens, and then the light illuminates the sample down here. So all of this, I'm sure, generates quite a bit of heat down here on the sample. So remember, Robert Hooke was able to look at cork, and he discovered cells, and he named them cells. Over here we have Leeuwenhoek's microscope, which of course is handheld, much smaller. You put your sample right here at the end of this little needle. You hold it up to the light, and you look through this metal plate, and there's a little lens embedded in the brass right here, and you're able to see your microscope, magnifying about 100 times. So pretty good equipment for the 1600s, but we have much better today. Modern microscopes fall into two categories. There are the microscopes that are powered by light, or your image is actually made by light. And then we have fancier microscopes now that use electron beams. Light microscopes have been around since the 1650s. They are still in use today. Those are the microscopes we'll be using in class. Electron microscopes were first invented in the 1930s and came into common usage in the 40s and 50s, and definitely still used today. There are advantages and disadvantages to both kinds of microscopes. All right, light microscopes, as the name implies, use light and a system of lenses to magnify and resolve an image. Okay, the light passes through or bounces off of a specimen. So if you're going to look at something under a light microscope, okay, say this blob of stuff, you got to do one or two things. You've got to shine light through it, okay, up into an eyepiece, or you've got to bounce light off of it. Okay, you got to get reflected light and interpret this as an image. That's the way a light microscope works. All right, light microscopes are limited by the wavelength of light. Believe it or not, light has a wavelength and a width. So if this is a beam of light, you can't see anything smaller than the width of the beam of the light. Okay, so from here to here, or from here to here, anything that's smaller than that, you can't see it with light, so you got to use something better. Light also generates heat, and bright lights can bleach out colors. So these are the main drawbacks of light microscopes. Light microscopes are limited in their magnification power to about 400x. Oh, wow, that's a horrendous typo up here. Forgive that. If you look up here, that's electron microscope. Why is it you never see these things until you start recording a video about it? Okay, electron microscopes don't use light. They use a beam of electrons and magnets instead of glass lenses to focus a beam of electrons either onto or through a sample. And just like with light, the electrons have to pass through it or bounce off of it. Electron microscopes are much more powerful. They can magnify up to 250 times, which is 250,000 times larger. And the downside is the specimens have to be cut very, very thin. They have to be sliced incredibly thin, or they have to be plated with a metal like gold to make them shiny so the electrons bounce off of them. But if you can handle these two problems down here, the payoff is you get enormous magnification powers. All right. Here's a picture of a light microscope similar to the ones we're going to be using in class. Okay, you'll notice there is an eyepiece up here where you put your face. This is a binocular one, so you can look through with both eyes at the same time. The image of what you're looking at is right here on the stage. So the light is coming up from the light source, okay, through some equipment down here to focus it, through a hole in the stage, through your sample, and up through a lens right here, which is the objective lens, then through some mirrors and out through the eyepiece into your eyes. Okay, so the light is actually being bent kind of around a corner here by a mirror. And light microscopes like this is a compound light microscope. Okay, it's called a compound light microscope because you can choose 1, 2, 3, 4 different magnifications here with different objective lenses. This whole nose piece here rotates, so you can choose how much you want to magnify what you're looking at. So that makes it a compound microscope. And you guys need to know how to calculate total magnification. Total magnification is a multiplication. You're going to be multiplying two numbers together to get your answer. Now, let's say the eyepiece is 10x power and the objective is 20x power. All right, we're going to multiply those two numbers together and get 200x. So that is our total magnification. It is not 30x. Okay, don't add, we multiply. Okay, electron microscopes come in two kinds. There are scanning electron microscopes, or SEMs, which bounce electrons off of metal plating that's been applied to whatever it is you're looking at. So you actually give them a mirror finished with usually gold. And these are very expensive machines, but the payoff is you get very cool three-dimensional pictures. For example, this is a scanning electron micrograph of some pollen grains from different species of plants. You can see there's amazing three-dimensional detail possible from these pictures. Here's a scanning electron micrograph of an ant head. You can see the ant's compound eyes. You can see the very complicated exoskeleton and antennae that it possesses. Again, very high resolution, very clear imagery in 3 dimensions. And here's one of my favorites. These are Streptococcus bacteria. They are magnified 65,000 times, looking down here at the key at the bottom. Each of those little circles is a bacterium, one single prokaryote cell. Notice how you get a 3D effect though. Not a whole lot to see, just a whole lot of bacteria. Now, transmission electron microscopes, or TEMs, produce a different type of picture. Here you have to pass electron beams through a very thinly sliced sample of whatever it is you're looking at. And you can get huge magnification with this equipment. It's expensive. It's not something you normally see in high schools. Most colleges have them though, and if you take the class, you can actually play around with electron microscopes. They produce interesting but flat two-dimensional images. This picture right here is a cross-section of part of a cell I'm sure everybody's familiar with, a sperm cell. So if you cut through the tail or the flagellum of a sperm cell, this is what's inside it. They have these little bundles, or a circle of pairs of microtubules with 2 in the middle. So there are 9 bundles plus 2 in the middle. So this is called the 9 plus 2 arrangement, and it's what makes flagellas move. Okay, but scientists did not know this until they were able to get a microscope that was powerful enough to magnify this much, to be able to see something this small. All right. In class, we're going to be practicing using microscopes by making wet mount slides, and wet mount slides are just what you think. They use water. That's why they're called wet mount. And if you remember, water has amazing surface tension, so we can use that property of water to our advantage. These are very easy to make slides. They will dry out, so they're temporary, but the good thing is if there's something swimming around in the water, you can see it while it's alive. All right. Wet mount slides have some basic parts. You have a bigger glass slide. Okay, this is what am I doing here? This is a glass slide. Okay, you're going to put your specimen, which you're going to look at, on top of the glass slide, and you're going to put a drop of water on it. Okay, so this light blue here represents water. And then on top of it, you're going to put a cover slip. And what happens when the cover slip touches the water is the surface tension of the water is going to pull the cover slip flat against the glass slide and flatten your specimen out. So the beauty of a wet mount slide is you're using the surface tension of the water to flatten out the specimen, because the flatter your specimen is, the clearer you're going to be able to focus it. All right, and we're going to look at a couple different things in lab there. This is a wet mount slide. All right, thanks for listening. That's the end of the video cast.